Okay, let's talk through the notes that you're going to use for the Krebs cycle. So based on where we're at right now, hopefully, recording this on Monday night, we have gone through all of glycolysis, and we are thinking about the movement towards the next step, which would be the Krebs cycle. And that's going to be our focus of our notes today. So just as a review, in glycolysis, we started with a glucose molecule, and that glucose molecule is C6H12O6. And when we got done with this, we ended up splitting that sugar, and we got two pyruvates. And with those pyruvates, there's still some stored energy in there. We've got each of those pyruvates is technically 3 carbons long. So basically, we've just taken glucose and we've turned it into two smaller sugars. When we do that, we did make some NADH that we talked about the other day. And NADH is an electron shuttle that's going to help us think of it as a piggy bank or a battery that's going to be able to power something later on. And that'll come up maybe tomorrow in lecture. And we also got a little bit of ATP out of the process, enough to run some basic systems in a low energy situation. It's not going to be something that's going to be able to power a multicellular organism, but it'll be enough to power something very small, maybe a single cell like a bacteria. So very primitive in nature, very basic. Glycolysis works for everything. The Krebs cycle, things get a little bit funkier. We're going to have to transition. We're going to have to start using an organelle to make this happen. With organelles, we're going to need some more membranes. We're going to need an increase of surface area, and we definitely need oxygen. So oxygen has to be present in order to move into the Krebs cycle. So let's start taking a look. So some of the products of glycolysis are going to be the reactants, and I apologize for the background noise. I forgot to turn that off before we got started, so we're just going to leave it there. Laugh it up, I guess, here, right? Um, Glycolysis, some of the products of glycolysis are going to be the reactants for the Krebs cycle, specifically pyruvate. That 3-carbon sugar that we had is going to be your starting piece for the Krebs cycle. Now, it's important to note that as we go through this little mini lecture via online recording, feel free to tell whoever the substitute is to pause it and rewind if you didn't catch all the notes and weren't able to write them. So take your time with this by all means. All right, so pyruvate has more energy to yield, as we said here, and we're going to strip off or oxidize those carbon sugars that are there. So remember, oxidize means to lose electrons. And I'm hoping that I say this on our lecture on Wednesday, but once again, I'm recording something that's after something that you're going to hopefully get on Wednesday. So hopefully I remember to say this, but the way to remember oxidize is like Leo the lion goes grrr, rawr, right? So loss of electrons is oxidize, and gain of electrons is reduction. So since we're stripping off electrons, we're going to oxidize. So the 3C pyruvates are going to lose their electrons, and we're only going to be able to do this if oxygen is available. So there's going to be um, some different types of reactions that are going to happen here in the Krebs cycle. There's going to be a lot of oxidation that happens, but we will see where there is a loss, there's going to be a gain in something else. So enzymes of the Krebs cycle are going to complete this full oxidation. That's the big goal here, right? The Krebs cycle's big goal is to fully oxidize whatever sugars we have left. And the result of the sugar is going to be it's going to turn into CO2. So anytime that you see CO2, we'll see some patterns here in this scenario as well. So whenever we lose CO2, the carbon chain, if we're going to draw it out like little circles, right? If we have a 3-carbon chain and we release the CO2, then that chain is just going to shrink in size. Now, I say this loosely that we're going to oxidize, but the goal when we oxidize is to make more shuttles. So we're going to end up making NADH. We're going to make this other molecule called FADH2, and you'll see all these coming up very shortly. All right. Woo! Woo! Listen to that. And you have this diagram off to the right. This is a fantastic diagram that's going to help you out, and we'll piece it together and we'll do a little mini poster. Hopefully you've already started it, but we'll get some more time with working on all that stuff so that you see how all this stuff is interconnected. So you can see that some of this stuff from glycolysis feeds right into the Krebs cycle, but also some of the things that we made from pyruvate, like the NADH, are going to move onward. They're going to jump, kind of like chutes and ladders. If you ever played that game, that was awesome, right? You climb all the ladder. Ooh, I got a ladder all the way up to the top, and then you hit the slide and you come all the way back down to the beginning, and you suck and you lose, right? Yeah. So I love playing that game. It's a total crapshoot as to whether or not you're going to win. Regardless, we're going to shoot all those NADHs out here to the electron transport chain, and we're going to do some massive amount of ATP harnessing, much more so than we get with substrate level phosphorylation. All right, so mitochondria structure. Let's keep on moving, shall we? Form is going to fit the function. So we're going to have this double membrane energy harvesting organelle. Wowza. You already know some amazing things about the mitochondria. Hopefully you can rattle those off. That's the extra ribosomes that they have. DNA allow them to reproduce. Now that mitochondria is fantastic. There it is, right? So that's a transmission electron microscope. It allows you to see super duper 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 small things by using electrons as opposed to visible light. Visible light doesn't have the ability to focus beyond the wavelength constraints that light has. Electron doesn't hold that, so it can actually see things better. So the outside of the mitochondria is smooth, and the inside is highly folded. Remember, anytime you see this highly folded, that means that we're increasing surface area. And that surface area increase is going to help us. And that surface area, sorry, I just wrote right over that, is that inner fold is called the cristae. So off to the right, I guess, this would be a great time to draw a mitochondria and label its parts. So we're going to label that anatomy, and that anatomy is going to help us figure out the physiology of how that mitochondria works. So do make sure that you have an outer membrane label, an inner membrane, and that inner membrane is called the cristae. So those are one and the same. We have this inner membrane space, and then deep down inside, this extra space in here is called the matrix, right? So make sure you take that red pill and go down the rabbit hole, right? As Morpheus would say to Neo. Go into the matrix, and that matrix area is going to be a special place where we're going to do different reactions than we do either in the inner membrane or in that inner membrane space. So we'll see that separation a little bit more tomorrow. So make sure you write down all those things. So the inner membrane space, the matrix, right? And then we have all that awesome DNA ribosome component that is in there as well. All right, so that is the mitochondria's structure, and that structure is going to yield its function. One of the biggest things, like I said, is this high surface area. Now, it's important to note that that cell membrane is very much like we studied in the last unit, so it's very complex. So anytime you see a cell membrane, you're going to see these little things, right? In your head, you don't have to shout this out. Those little things are the, that's right, phospholipids, and the phospholipids provide a barrier, so they're going to separate outside from inside. And then in those little barrier components, we also have little doorways. Yes, and you know those doorways, those are those integral proteins. And those integral proteins are going to provide a variety of different functions that we'll see, from moving electrons to being a facilitated transport component. And some of them get really kind of funky where they act like kind of like a pepper mill, right? I don't know, going out to dinner and they ask you for fresh crushed pepper. That's how this one integral protein is going to work when we see it in the electron transport chain. Onward we go, right? And we have some enzymes and whatnot, and we'll see those. There's another mitochondria. You can see some of them are actually dividing there. Kind of cool. All right, so oxidation of pyruvate. 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 We're going to be looking at how pyruvate loses its electrons in the first part. Now, in some textbooks and in some areas, we would call this step 2. I consider this part of the Krebs cycle. If you're trying to draw a generic picture, a cycle would be circular. This would be glycolysis over here, and then we have this like little transitioning staging area before you get into the circle. That's going to be this oxidation of pyruvate. So pyruvate is going to enter the mitochondrial matrix. So here's your generic background cristae. You can see that out here would be the cytosol, or the cytoplasm. We're going to transport that pyruvate into the matrix area, so we're going to have to cross the outer membrane, the inner membrane space, the cristae, and then we'll be in here for the Krebs cycle, right? So that's in the matrix, the deep down. See, the pyruvate is going to turn into this molecule called acetyl-CoA, and when it does so, it releases CO2. Now, I don't know if you remember 2 slides ago, I said that there's going to be some patterns. Whenever you see CO2 being released, you're going to see a change in the carbon chain. So remember, pyruvate is a 3-carbon, so the chain of 3 C's here. If we release CO2, that gives us a pattern recognition to know that acetyl-CoA is actually only 2 carbons. All right, so 3-step oxidation. So we're going to multiply this by 2. Why 2, Mr. Grant? Because we started with a glucose, if you remember. And when we split glucose, we got 2 pyruvate. So this gets done twice from the original 1 glucose that we started our story with. CO2, 1 for each time that we're going through this. And some, there's your oink oink piggy bank. So we're going to oxidize pyruvate. So that means that pyruvate loses the electrons. Who gains the electrons? No more than NAD, or NAD plus for more preciseness. And when it gains that electrons, it becomes NADH. Now, reason a piggy bank is fantastic, or the idea of maybe a battery, if that works more consistently for you and your knowledge purposes, Duracell, copper top, whatever, it's stored energy, right? It's stored, it's something that we can get later on, right? And so we get these 2 NADHs as well, right? So producing the 2 acetyl-CoAs, and the 2 acetyl-CoAs are going to be things that up here in this circle are going to feed in to the Krebs cycle. All right, so onward we go. Why are we going to get 2 turns of the Krebs cycle? As Dr. Penguin has said, is going to be exhaled. That's waste product. Okay, what is waste to us, though? We'll find out that is fantastic stuff for a plant. But word it goes. The sound effects are just fantastic, are they not? All right, so this is just summarizing. This is the same one that's out of your textbook. So you can see that we have transport protein here in that outer membrane. That transport protein is only permeable if O2 is present. If O2 is not present, the pyruvate is not going to be getting in. So O2 has to be present in order to transition inward. As you can see, we're going to release CO2. We're going to gain some NADH. And then, not that it's super important, but we bind in with this coenzyme A, which is a complex structure. And you can see that the resulting 2-carbon molecule is left over. Why 2? Because that CO2 got released as we push through. How many times does this go through? 2. That's right. Because glucose gets split in half before we start up this whole process. Onward we go. All right, so this is the guy that you can hate if you hate the Krebs cycle, also known as the citric acid cycle, and we'll show you why it is the citric acid cycle in a little bit. So let's talk a little bit about the Krebs cycle. And here's a little bit of history behind Hans Krebs and his contribution. So he figured out that this is a multi-step pathway of catabolism, right? Catabolism, I know maybe you've heard of metabolism, meta. Meta means middle, or balance between the two. There's catabolic and there's anabolic. Catabolic means to break down. So he found out the breakdown process that is involved with glycolysis, okay? And all this ends up making a heck of a lot of sense when you start looking at the timeline of how all this stuff works. And this is like it's saying here, we really didn't start seeing Krebs cycle development and the understanding of it until we really understood glycolysis. And more importantly, bacteria can only do glycolysis, right? Why is that? Well, they don't have mitochondria. So it makes a lot of sense that citric acid cycle didn't develop until after we started getting some eukaryotic organisms that were participating together in a symbiotic relationship. All right, and then we end up getting oxygen much later on because of photosynthesis. So onward we go. let's look at the process. So I'm going to turn this wheel. I'm going to run through some stuff here. You're going to write some things down. We're going to pick up some patterns, and we'll probably be able to finish this off in the next couple minutes here. So let's make sure we watch and count the carbons. So here's what we just talked about. Right, this is the preparatory stage. That's that little spur component to the circle that I was talking about. So there's pyruvate, there's acetyl-CoA. What is guaranteed to be released here? That's right, CO2, and you'll see that pop up here in a second. So oxidation of sugar is the big thing with the Krebs cycle. Remember, oxidation is a loss of electrons. 6 carbon, what? Where did 6 carbons come from? Well, if you notice, there's 4 carbons that are already in there. So 4 plus 2 makes 6. So when we start our cycle, we're going to start the beginning thinking part of 6. So there's a little enzyme in here that helps with the attachment of that acetyl-CoA with that 4 carbon that's there to make the 6. If we go around, you can see, oh, 6 to 5. What's the pattern? You can already know. How does the carbon chain length compare to the CO2 produced? You should know this now. So there goes the CO2. And back to 4. It's going to happen twice. So if you think about it, our original formula, C6H12O6 plus 6O2, we haven't even seen this yet, turns into 6CO2. 6, where did it all come from? We released 1 up here, and then 2 here. But that's only 3. Oh, remember that glucose got split in half as we feed through. So there is the account of all 6 of your CO2s. And then what we have not talked about is the water, and all that water stuff is going to show up more tomorrow. So count the electron carriers. This is another piece that you need to write down and pick up on patternness. So there's that citrate, or the citric acid water. You get some oink oink, some more oinkers, and oh, big money. We get a little bit of ATP out of this process, but it's so teeny tiny that it's still substrate level phosphorylation. Wow, that sounds so fancy, doesn't it? I know you want to go home tonight and you're going to talk about phosphorylation. Hey, when mom asked at dinner table, or dad asked at dinner table, what'd you learn about today? Well, I learned about a little bit of substrate level phosphorylation. Wow, I'm pretty sure they'll stop talking to you right there. So if you want some peace and quiet, start talking substrate level phosphorylation. But this is a teeny tiny amount. You're only getting 1 from 1 turn, so it'll be 2 total from the original glucose. Check it. We still got some more piggy banks to oink oink, a new piggy bank. This one is FAD, more specifically FADH2. FADH2 is a new molecule. It only really shows up in this Krebs cycle here. And we get NADH. So you're going to get 2 more NADH and FADH2 and 1 more NADH. So we got 3 NADHs and 1 FADH2 from 1 turn of the Krebs cycle. So multiply all that by 2, and that's the total you get from 1 glucose. We are almost done. Nice job hanging in. Over some more details here. Okay, so this is what the heck is going to show up on your test, and you might look at it and go, what the, come on, seriously? But it's all about pattern recognition. That's what this is all about. So reduction of electron carriers, how many get made, and what enzymes do it. So review. What type of enzyme is going to be making NADH? Hopefully you remember it is dehydrogenase, right? Dehydrogenases are going to be working with the NADHs. So from 1 turn, we have that little spur. We're going to get 1 up here, so that'll be an NADH up here. Okay? So it's that pyruvate to acetyl-CoA. You'll get an NADH there. You get 1 here. You get 1 here. You get 1 here. So from the original start, we're going to get 4 NADHs. Okay? But remember that we turn this twice, so they'll get multiplied by 2. So we'll actually get 8 NADHs from this Krebs cycle. How many FADH2s do we get? 1 here multiplied by 2, you're going to get 2 FADH2s. So remember, these are piggy banks. This is stored energy. We haven't even got the energy out of it. And how many junky little ATPs have we gotten? 1. And we got 1 ATP times 2. So we've gotten 2 ATPs. So look at all the stuff that you've learned so far. We've done all these steps. And what do you got to account for it? 4 ATP. We've done all of glycolysis. We've done all of Krebs cycle. And you've got 4 stinking ATP. That is it. But think of all the NADH that you have. If you think about it, glycolysis got you 2. So at this point right now, you have 10 NADHs. That's a lot of NADHs. That's a lot of stored energy in the piggy bank. And you got 2 more FADH2s. So you've got 10 NADHs and 2 FADH2s. A lot of stored energy that's going to power pumps. And pump power equals lots of ATP. So where do the carriers of our electron H pluses go next? And hopefully you have a little bit of background knowledge. The next step in our story, choo-choo, hop aboard. It's the electron transport chain. The ETC, that's where the electron transport chain is going to take our, wait for it, electrons, right? Electron transport chain is going to take our electrons. Where are the electrons right now? Oink, oink, in the piggy. So we have this as our next piece. And that electron transport chain is going to be the thing that's going to jack up the amount of ATP that we can get. And you are almost done with learning the cellular respiration process of awesomeness. Okay, so pattern recognition is huge here. We're going to review this stuff more. Hopefully you got enough out of this. I hope it didn't make you go cross-eyed, and if it did, have your lab partner smack you in the back of the head. That tends to help straighten the eyes. We will see you tomorrow. Thanks for playing, folks, and science on.